For all of our attendees, I'm thrilled to introduce our panelists for this very exciting session, the role of compounding pharmacy in the future of clinical management of diseases. So this is going to be a free-flowing discussion. We have some objectives which are going to basically function as just some jumping off points for the discussion. And we'll start that with the first point that we thought we'd talk about is to talk about demonstrating how we define the way clinical management of diseases are done and the role that compounding pharmacies can play. Do you guys want to just jump in on this? How do you want to, who wants to start?
Well, Vivian, I can start if you want me to.
Yeah, thank you.
Okay. So I'm Dave Kisor. I'm a pharmacist by training and really focused on pharmacogenomics. Follow Scott a lot about pharmacogenomics, but from my aspect, we're getting to the area of precision medicine, the era of precision medicine, where we want to really refine a patient's medications. And one of the aspects that I see with compounding pharmacy is the ability to get precise doses. In pharmacogenomics, where we're looking at the patient's response to medications based on their genetics, we now have over 25 guidelines that are available to help folks, make clinical decisions, incorporating genetics into the rest of the information they have. But oftentimes we can run into the case where we have specific dosing requirements that are not available in standard strengths. And this is where compounding pharmacy can really come into play, I believe. I think it's really going to be the key to getting that precision medicine. For instance, we might have somebody who is what we call an ultra-rapid metabolizer, needing a much higher dose of a medication. And therefore, that strength may not be available. And can a compounding pharmacy provide that connection? So I think it's really, really important from that aspect.
Mm-hmm. I agree, David. I was thinking about, when I heard the question, I was thinking about kind of historically and how far it's come. So I've been around for a while. I'm a pharmacist by training. For the last 10 years or so, I've been dealing really with data curation, both of drugs and metrics that we were doing for adherence and compliance, et cetera, in a previous part of my career. But, when you think about it over time, how rapidly everything was on paper until really very recently. And so the ability to capture a lot of that information electronically and then be able to use it now, it's been really important. But even on the pharmacy side, when I was dispensing, very first dispensing was on paper and we typed labels. And so I know I'm dating myself a little bit, but just in my career, we've covered a lot of ground in a relatively short amount of time, and being able to capture that data to apply toward, customizing therapy for people has been really, there's some catching up to do. It's kind of doing, spurts over. But.
Yeah, I think, when you speak about the data that we have and trying to pull that all into a decision support kind of tool, wherever that decision support is, whether it's in the pharmacy or, with the patient interface in other settings, it's really become important. I mean, that's what we're moving to. I think something like 89% of clinicians now use electronic health records. And so, we need this interoperability to pull in data from different systems and also from databases, right? And so I think it's in the theoretical realm, we can talk about pharmacogenomics and we have 25 guidelines plus, and it's being applied in a lot of places now. But, and I know, Scott, you've mentioned this before, and I'm going to steal this from you. You talked about, so the real endpoint is getting this kind of printer idea, right? You've heard of these 3D printers, and Scott's mentioned this a couple of times, but I really think that that's where the compounding pharmacy will complete that circle, right? Being able to get that actual dosage form that's precise.
Yeah. Yeah. we had a lot of discussions about the future of pharmacy around Cardinal and specifically in Fuse, where I was working about what would that look like in the future. Is it compounding tablets, and there's real life examples of that now that I think we're all aware of, and then printing into the adhesive layer of a patch or something. And there's of course, I think as Tom pointed out in the earlier in our Connect last week, there's regulatory and there's GMP type considerations there. But yeah, getting down to that drug, that dose for that patient is really
and there's a number of resources out there now. I guess from the federal side of things, the Food and Drug Administration has provided a table of pharmacogenetic association. That's the real connection with using pharmacogenomics, because it's not often found in the drug labels, right, in the labeling. And so you need to depend on other resources, and the FDA has that resource available now.
One of the things that there was an oncologist in New York who I think was doing a study for Flatiron, where he was trying to discover a lot of unstructured data within notes fields and within fields within the EHR, because the EHRs really, there's a lot of things that get bolted onto them, but functionally they're, I think everybody agrees that they're that can be difficult to work in. And so there's data stashed in notes fields and labs and things that aren't in a discrete field, that aren't really discoverable. And so there's unstructured data that's out there that that is necessary for this kind of work too.
Yeah, that's a great point. We've got the situation where with the early genesis of pharmacogenomics, laboratories have been providing reports that are essentially PDFs. And so they'll be attached to some electronic health record. But here you're talking about, maybe 45, 50, 60 pages of information that needs to be boiled down into discrete fields like you're talking about, Scott, right? And so they can not only be, used for the individual patient, but also for data aggregation, right? And putting together information for the health system. So, from the compounding pharmacy side of things, I know there were pharmacists in Vermont that worked very diligently to get access to the electronic health records. So from different entities, so they had that information in their compounding pharmacy to make the same kind of clinical decisions that others would make. So that's really important. I think right now, with Epic and Cerner, maybe the two biggest, at least well-known names as far as EHRs go, they don't provide a standard module for genetic data or connection with pharmacies, right? And so these are being built in individual modules by different entities, and of course they're different everywhere you go, right? So the interoperability of those is minimal to none, and we've got to get patients to the point where they know that their data is theirs and not a certain entity's, and it's untethered, so that pharmacies have access to this data.
Yeah. I mean, I'm looking at a pharmacogenomic report right here, which happens to be mine, and it's 20 pages long. And so we're reducing 20 pages to some actionable data, and it's color coded with red, green, and yellow smiley faces at the drug level, but it's a lot of pages to look through to see what I could or shouldn't be prescribed, or if there should be a dose adjustment or even a therapy adjustment. It's hard to look through.
Yeah, I think that the regulatory agencies have been really putting their thumb down on pharmacogenomics because of the lack of standardization and what information is being given to clinicians and patients. Now, it's not that they have their thumb on it now as much as they used to. This was early on when some laboratories came onto the market and were charging exorbitant amounts for PGX testing. That's changed, completely now. Two big things have happened in the last year and a half. The first was that in 2020, and I guess a little bit over a year, in February, the FDA published that table of pharmacogenetic associations. That's the first thing. The second thing is, as far as coverage for pharmacogenetics, the Medicare administrative contractors in molecular diagnostics, those contractors, there's 4 of them, had a coverage determination that is more broadly covered pharmacogenomics, especially in behavioral health. So in psych and depression especially, but broader yet. So that's good news for pharmacies. And again, I think the whole idea of being able to compound that precise dose based on evidence-based guidelines is really great. So.
Yeah, agreed. Lee and Tom, are you able to hear us?
Yes, I can. I'm in now. Thank you so much.
Okay, great.
I didn't do it.
I love it.
So we were just in a discussion about the current state, kind of general idea of where disease states are being managed, and some nuances and some new things that were going on. So.
Yeah.
I was going to say, I don't know if you guys got to this, but I thought we'd also talk about how compounding pharmacies can deliver novel medicines, including combinations with the ability to fine-tune the dose to specific patients.
Yeah, we did talk about that a little bit, but there's certainly more discussion that maybe Tom and Lee can add to in that kind of thing.
Yeah, my concern is the quality aspect of it. I think there's no doubt that this precision medicine is the future, has a future clinical utility for the patient. And how do you, I think, is the big question and conundrum, how do you compound a unique product on a unique patient? Already the question's up, or from a quality perspective, and you want to ensure you have a safe and effective product given to the patient. So how do you maintain that potency and consider the different excipients and the dosage forms, as well as even the container closure ability? So there's a lot of information and thought process that needs to go into that process, and even maybe change the way we're approaching, or approach the thinking toward it from a practical quality perspective.
And it's in, actually, as we the more of the individualized based medicines, things like CAR T, where you have individual doses that may be $1 million more. The interesting thing is those are really based more for individual individuals as they move forward, because you're really hacking, for lack of a better term, the RNA or specific DNA of an individual. And so you really are basing your medication on 1 or 2 individuals that are very, very similar. Those are really geared towards individualized. It's hard to make a patient where your audience is one. And so to become more individualized, particularly in genetic medication, I think you'll see even more of a need for individualized medication in pharmacy. I think we're starting to see that more with the recognition by providers and practitioners that there really is a unique need for these types of medications. Even things like water pills, things like lisinoprils or hydrochlorothiazide, or the need between a cardiac patient versus a PEDS patient, for example, or diabetes, those doses can dramatically differ. As a pharmacist, I think one of the things that we is that unique dosing, and that will become that much more important as we move forward.
There in Europe, as I'm thinking about it, I think maybe in the Netherlands, either they have done or are doing a combination printed tablet with 4 different medications in it. Is that ringing bells here? But I didn't know, Tom, have you heard that? Impact of quality, from some of this work that's been going on overseas.
David, some of the sound, Lee, we can barely hear what you were saying, and even Scott, it's a little blurry, but I think you were talking about some of the work being done in the Netherlands and printed type medication. And I think that the approach is, it's almost, I think of like the automated machines in the hospitals now, where they're validating the process, and they're validating the sterility, and they're validating the delivery mechanisms. And then once you validate that process, more like the GMP approach, then that they perhaps could be integrated or flow into a real life practical, compounding world, perhaps. But there's a long ways to go from that perspective. Is that what you're talking about, Scott?
Yeah, I just, I know they're doing that kind of work over there, but I haven't read about it for a while, and I didn't know if they were finding on the therapeutic side or, on the outcome side, whether this, combination of four different drugs is showing any of these quality problems or anything.
David, are you aware of any compounding pharmacist that is actually testing the patient and then compounding that specifically and then doing quality control on it, all three aspects of it in a practical real life situation today?
I know of compounding pharmacies in Indiana, in the Fort Wayne area that are doing that. They're adding in the pharmacogenomics side of things, though, to be more precise. I would say also, relative to what Scott and what Tom were talking about with combining medications and making dosage forms, it really, on the patient side, comes to improving adherence, right? And being able to, just be more adherent to these different regimens. And there's ways to do that. I think we're at the start of this whole process of really improving patient adherence because of pharmacogenomics also.
And David, I would agree with you that we're at the very beginning. I don't know if you guys can hear me better now. I apologize for a minute.
Yes, we can. Thank you.
Perfect. Like I was saying a minute ago, we're really aware of three particular disease states where this is really starting to happen. One is in the BHRT space. Think of it like testosterones, right? Where you have an individualized clinic that will be doing the laboratory work and then can prepare the compounded medication directly for that individual patient needs. And the needs, particularly in the hormone space, can be dramatic differently. It could be whether it be somebody is going through a change, right? Whether it's for prostate use for testosterone, or whether or not it's for an individual male who just needs it because they're reducing their testosterone, for example. And so you can think of the different doses that all three of those would need if you were doing a female-to-male change, whether you were doing a prostate change, or for a male who just needs it themselves. How those would be needed for the individualized aspects, and it could be tied to the lab quite easily. Another one, and I think it's probably the most relevant, is in that CAR T space. So you'll have a medication that's $1 million easily, right? Where it's really geared for an individual patient or maybe a family where it's one or two an audience of. It's not really made for CGMP type production because you're really producing it for one or two individuals. But yet you'll have a contract with the PBM that'll say, look, if it doesn't work, right, we'll only pay you half, but if it works, we're going to pay you the full thing. So you need somebody who's got that type of production at a very sophisticated compounding pharmacy who can help make that medication and prepare it, that it's potentially $1 million lying on the line. Mm-hmm. And so I would say the other that we tend to see is in the onc space, the oncology space. Think of it like breast cancers where you have HER receptor positive, for example, where you'd have a particular medication that's needed for a very specific breast cancer, right? Again, those are individualized needs. Obviously, the IVs usually are done as a compounded, whether they're in a hospital, whether they're in a pharmacy. And you're starting to see more and more of those needs where you're going through a specialty pharmacy with a special PBM contract.
It's interesting. I think of very good comments on Lee on the two aspects on that. First, the BHRT. That reminds me the method or the approach is more like the old therapeutic drug monitoring, where they're measuring a level of testosterone, and then they're saying, okay, based upon this outcome or this true marker of the level of testosterone, then we're going to treat you according to that dosage. So you have that individualized patient, you have that result, and then you have that treatment. That's different, whereas the pharmacogenomics I'm seeing involved more in the oncology space, where they're looking at that HER, the different and the specific slow or fast metabolizers. What are the markers in relationship to that treatment? So they're almost doing the genetic test prior to the drug, just based upon your genetic and dosing based upon your genetics, where the former they're doing that drug levels or your natural levels of testosterone. But I definitely think it's early on, and I think there's so much opportunity for the CAR T cells, especially talking about personalized. That's your own cells.
Exactly.
Yeah, pharmacogenomics is taking a more predictive is probably not the right word, but it's we're gonna assess what the therapy likely is going in that direction versus taking blood samples and into what would be a dose to give that is for that patient. So
yeah, right.
A population-based type parameter versus individual back toward
Right, and Scott, I think what you're bringing up is really important too, and something I want to talk about what Tom said too. So Scott, from the standpoint of being reactionary versus being preemptive, right? That pharmacogenomics allows you to be preemptive and have some prediction of what that patient's going to look like as far as handling medication. But right now we're in the reactive stage, right? I mean, you give a medication, something's not right. Maybe it's inefficacy or it's an adverse drug reaction. And then you try to say, okay, why is it happening? And then you go back and check in the genetics and you see it, right? So we need to get to the preemptive standpoint. And Tom, relative to what you were speaking about, I think it's really important, as small as a study may be, that these studies are done in the compounding world, where the aspects of precision are put into place and outcomes are measured. Because you're not going to have these kind of studies from a clinical development standpoint. A couple of reasons. One is that some of the genetic variations we see in patients are so rare that you're not going to be able to recruit enough patients into a clinical study to have an N that will be powered the study enough. And secondly, some of the the outcomes are adverse reaction based, and so you're unethical if you're going to randomize somebody to an outcome that's an adverse event. So some really great points, Scott and Tom. Yeah.
Well, and David, I would echo that, right? As we move farther along, one of the things that we're seeing, not to throw shade at anybody, right? But if you look at the NASEM reports or you look at FDA reports, is the requirement for additional clinical information. The problem becomes is as we get into more of this individualized personal medicine, how do you do a study with an N of 1 or an N of 2, right? You really can't. And so you can't shoehorn it into the need like you would in the situation for the same type of clinical information and say, look, we are going to require that when you're really trying to individualize this to a need where you couldn't do that clinical trial even if you wanted to.
I think this is some excellent comments because of all the issues. I think you're right on target of this bioidentical hormone replacement therapy, this BHRT. And Dave, your thought process of looking at the outcome, that's exactly what we need to do more of. We look at the quality. Obviously, I have an inherent bias toward the practicality of the formulations or pharmaceutics aspect of it. But you're looking at, is there a patient outcome and a positive response without an adverse reaction? I think a more focus needs to occur on that. But these kind of discussions that we're having here today, because you have 4 individuals from all different perspectives coming together for really that positive outcomes for that patient. I think, Lee, you hit the nail on the head with that, the BHRT. I think that's a big issue. That's a big component of compounders today. And then definitely it's being looked at and addressed, but yet you have literally millions of patients across the United States that are utilizing that, and that question is being looked at. Is this what needs to be done with it? How to do it? And I think sometimes we forget about the patient and the patient's outcomes. So I think those are excellent comments, David and Lee and Scott.
And Lee, to your point, there's a paper that came out, and I'll see if I can somehow get this to everybody through this venue afterwards. But the study was called the N of 1, and it's a really interesting look at what you're talking about.
No, I appreciate that, and I think it's extremely relevant, particularly now, as again, nothing wrong or right as to what was being said, just the requirement or request for additional information. It's important to remember where we're at, and as unique as we're trying to make this, particularly in the individualized side and personalized side, you really can't make it into a clinical trial. And I think that becomes the crux of the issue. FDA themselves are so used to, and most healthcare providers are the same way, so used to what has been done over the last 50 to 100 years of large clinical trials, and it doesn't fit itself there. To me, I think that's where we're really heading as we start to think about this more, right, as a holistic approach as a healthcare provider, right? If I take the pharmacy hat on, the advocacy hat off, is you're wanting to have these pharmacies and providers and labs really work together so that way they're tied together. So somebody can walk in, get a blood test, see what it's done, walk over to the provider and say, this is what it means, and walk over to the pharmacy so they get an individualized patient treatment for that exact need. And as we get closer and closer to that, right, it rubs with what we've done over the last 50 to 100 years.
Yeah, I think that's a very thoughtful perspective on it. I mean, and then take it even a step further as we're sitting here fantasizing about the future. I mean, I think this whole topic, this was an excellent thing that the Pharma Salon pulled together, this role of compounding pharmacists in the future of clinical management. Take what Scott started out the discussion with, maybe a vending machine, of going in, seeing it, and then printing, doing the test, genetic test, coming up with the medication, and then printing your drug.
Right.
I mean, that would almost be like space, like.
Mm-hmm.
I think
the future.
I do think that it's really important for compounding pharmacy organizations to present case reports, individual case reports, where there's outcomes information, because these bigger studies are not going to be done. Sponsors are not going to pay, as Lee was saying. Sponsors are not going to pay for more clinical studies once their drug's on the market, unless they see some kind of benefit, and they're not going to see it in this realm, right? So I think it's a great point.
Even if they do, you'll end up like, if you do, they'll end up like CAR-T, where the medications are $1 million in individual treatment.
Right. Yeah.
I love your recommendation, Diva, of doing more. I think sometimes the compounder pharmacists, they think they're not doing those clinical trials, and so maybe not, because often they're operating more in a micro section of their community. So it's more of a community area. It's meeting that state, that region or that rural type process. But it would be good to try to get a lot more literature or published in the peer review. And I think maybe the acceptance also would be better from that, to get greater case reports, which would aid in the education, as you were saying, that the NACE report's requesting more of that.
Mm-hmm. No, I agree.
I think from the payer side, there's a pragmatic approach that they take to these things. We're all aware that CAR-T is so expensive, but it's a risk benefit, it seems like, on their side to the payer, and not a risk benefit to the patient. And so that pragmatic approach to approval or not approval oftentimes rules when if we did have the data, we could, like you all are pointing out, if we did have some solid data we could point to in the form of case studies or larger clinical trials, I think that would carry a lot more influence on
Well, and I agree with you, right? It's somebody on the other side, right, who represents the pharmacies in the situation. That becomes the rub. Look, we have put ourselves in a position with some of the things that happened with skin creams over the last couple of years, right? Well, let's be honest. On the flip side, though, it shouldn't be that one disease state should be looked at as it representing the entire industry. And I almost wonder if it's not reverting back to, 10, 20 years ago to say, look, this is sort of like a capitated model. And so we've got to do what we've got to do. We got to figure out in a situation whether this individualized personal medication is beneficial-wise, right, risk versus benefit calculation, as compared to a PT or whatever the other type of treatment would be, surgery, etc., moving down that line. And I wonder, as we move forward, especially with trying to, let me say it this way, reduce the amount of drug spend that we have, that if we don't start to see some more of that beneficial calculations to say, okay, do these personalized treatments really mean more? And if we reduce the cost of them, instead of doing $10,000 a treatment, if it's $500 a treatment, but we have all the other lab costs that go with it, does that equal out as to a reduction in the amount of risk that we have as a company?
One of the things about lab costs from the pharmacogenomics standpoint is, right, it's a one-time test. And so it lasts that person the rest of their lifetime. If, of course, that test includes a broad enough panel that will cover certain medications they may be on at some point in time, right? If it's too narrow, you might have to go back and get tested again, right? Or at least if the samples are stored, have them run again. But no, I think that's a really big point, that it's a one-time test and costs have come way down for the test.
I think it's excellent. Even in oncology, it is commonly used. Even for pharmacoeconomics, just think if you're a slow metabolizer, you don't need that much. Instead of spending $100,000 for a drug, you're spending $10,000 for the dosage that you amount. So from that perspective, looking at it from the pharmacoeconomic perspective, but I'm thinking let's say you do that as a part of the regular regime for that patient, then you would know right up front, if you're a 2D6 metabolizer, slow metabolizer, you're going to need to alter that dosage form. No one is better. I think the compounder, let me reword it, the compounder is in a great position to truly personalize the first time you set it up, and then that individual retains his genetic profile throughout from his physician, and he can say, yes, we know we have a drug-drug interaction. We know I'm a slow metabolizer, I'm a fast metabolizer for your particular enzyme. And then they can direct the effect of the treatment. I think too often, we wait until there's an adverse event, and then we say, hey, I know I published I did a presentation back in, I think it was 2017 at the American Academy of Forensic Sciences. It was on the perfect storm. And it was an autism individual who had passed away. They were going to rule it as suicide, but in reality, he was a slow metabolizer, and he had the 2D6, and it was whey. Well, he was small, he was nonverbal. The doctor felt they needed to give him more, so it was the perfect storm. Excessive amounts, the weight was small, he was a slow metabolizer, and on behalf of that, they gave him an additional drug who had a drug-drug interaction that the physician was not aware of, the genetics the genetic issue.
Yeah, I think Lee and Tom, those comments are great from the standpoint of decreasing the amount of drug you need, right? I mean, that cost of that drug, it's not really benefiting the patient, right? It may harm the patient. I mean, there's a very famous case where there was a mom who, during the birthing process, had an episiotomy done. You know where I'm going, Tom? I see your head shaking. And the mom was given codeine with Tylenol, with acetaminophen, right? And she was an ultra-rapid metabolizer. She converted so much codeine to morphine. Every time she breastfed her baby, the baby got a dose of morphine, and the baby died of morphine toxicity. So, there's a lot of cases, but those are individual cases, and that's not a study you're going to do. You're not going to randomize people to a toxicity arm, right? So your points are really important, and the compounding pharmacists can do that. They have to follow that all the way through and document that.
But how do we move this idea into the future from a practical perspective? Because these are all great ideas, but how do you make it a reality where it can occur on a regular basis, which would benefit by lowering the cost of the drug, but also having better outcomes for that patient?
To me, it's the discussion that we've been having over the last 10 years as pharmacists. It's that we need to be more than just a vending machine, right? We need to be paid more than just licking stick and throwing out the drug in the situation. We need to be looked at more as a provider. And we need to be able to understand that we're part of a team with a holistic approach. And I completely agree with you, David. I think in this situation, I think as things move forward, I wouldn't be surprised if you don't start to see younger and younger individuals, particularly just as they're born even, have blood tests done from a pharmacogenomics side, so that way you'll understand it moving forward as part of their permanent medical record what somebody has, for example, and SIP proteins and all the other things that go along with that. And if we would have that, now all of a sudden the pharmacist as a provider can give a heck of a lot more valuable advice, and I think that you start to look at more as a provider status than you are as just somebody who's a vending machine to sell drugs.
I think another important thing, and sorry, Scott, were you going to say something? Okay.
No, go ahead, and then I'll follow.
Yeah, I think another important thing is to understand a recent ruling by the Food and Drug Administration relative to direct-to-consumer genetic testing. They have approved for direct clinical use the CYP2C19 gene information relative to 2 drugs so far, clopidogrel and citalopram, but more drugs will be coming. So think about this. For the first time now, you can have a patient send their sample to a lab or to a company who has a lab, works with them, get the information back that can directly be used to affect their therapy without having a healthcare provider or professional in that loop whatsoever. Now think about them with access to the compounding pharmacy or the pharmacist in general. And so that's a really important ruling. It really has opened the door, I think, to some things.
I agree. And I think the COVID situation has helped that, right? Because I think we're actually now starting to see even at-home COVID tests, for example, where they just give an authorization for one. So I think you're going to start to see more and more of that where individuals can take care of themselves at home and use that to walk into the provider more informed.
I think the COVID situation, Lee, has definitely illustrated the importance of the compounded pharmacy there. Now, I'm going to take another step from what David was saying, that you take that prescription, you take that information to that pharmacist. He needs to be understanding the educational IQ from the pharmacogenomics. But then if he does prepare a tablet or a capsule or a dosage unit, it needs to ensure that it's the right potency. So they have the processes in place that if you are delivering that it's not too much or too little.
Exactly.
I was thinking about even getting to the right drug quickest. And so you have situations where, particularly with antidepressants, and it's often you have to try one, and they either don't reach a therapeutic endpoint, and they don't have a toxicity, and many people will drop off of therapy. They're not going to receive any help, and it's getting to the right drug and the right dose as quickly as possible to remove all that variation and stuff. And there's a cost, of course, both to third-party payers, and so that's important too. We're not completely focused on adverse reactions, but people dropping off therapy because of not being able to find the right drug as fast as they could.
Yeah, I do think that as these different avenues to getting information relative to precision medicine are opened up, that the sponsors, the manufacturers, are not going to be able to respond in a manner that the individual compounding pharmacist can respond. And the FDA is going to want some data behind this, and that's why I think I'm not admittedly as familiar with the compounding pharmacy organizations, but I think that's something that they really should focus on in supporting their members and the community as a whole to document this, this aspect of compounding pharmacy with precision doses and then outcomes.
I 100% agree with you, right? As the chair of the 503B Trade Association, the issue becomes, like we were talking about a minute ago, how you do a clinical trial for one and the cost associated with it. That's why I'd love to be able to see that article you were talking about, right, to bring it full circle.
Okay.
That'll be interesting to read.
Mm-hmm.
I had that in my mind, Lee, but I wrote it down just now because I know I'm going to forget it. So.
We won't let you forget. I quoted that
that.
article, but I haven't read it. That is supposed to be a good article.
I don't know if you guys see, but there's a question. Are there technology solutions for automating patient outcomes review and documentation?
So I know that there are a number of companies that have built algorithms that take into account the information from technologies like DNA testing, but also lifestyle, environmental, other drugs. Let me give you an example. There may be data looking at potential drug interaction, but as Tom alluded to before, if somebody is already a slow or what we call a poor metabolizer, and you put a drug that inhibits metabolism on top of that, well, the patient's already a poor metabolizer and can't be knocked down any further, so it may not really be a drug interaction. And so these kind of algorithms are starting to put this together as to say what is really a drug interaction and what's not a drug interaction. And one of the issues, for instance, with the local coverage determination by Centers for Medicare and Medicaid Services relative to pharmacogenomics, they said in their document that they would be moving away from paying for these black box algorithms because the information of those algorithms are proprietary and not publicly available to be tested. Now, having said that, some companies have done their testing, have done studies, and have proved that their algorithms are working. So I hope that kind of answers the question about putting this information together, and outcomes fall into that algorithm in an iterative process, what worked and what didn't work.
Any other questions? Okay. I know you guys, we wanted to make sure that we reviewed all of the objectives. And I know you guys touched on everything, but just to review our last objective, and I don't know if you guys wanted to kind of just revisit this or talk about it a little bit more, is to examine the technical, regulatory, and logistical challenges compounding pharmacies need to consider. Like I said, I know it was all kind of wrapped up, but I just was wondering if there's anything else you wanted to add to that.
Sure, I can jump in real quick, Vivian. As somebody who works with 503As and 503Bs, right? I mean, one of the biggest issues that we have with 503Bs for the issues we've been talking about is when you have an individual need, you tend to have to make things from bulk, right? Because we're changing it from what's commercially available. In the 503B space, it's tough because the FDA has limited what you can do from bulk to the bulks list. The A's have a little bit differently, right? Because they can do things that have a USP monograph, and there's some other aspects that go along with that. But in the B space, we are limited to it just based on the individual clinical need, which really ties it back into, like we were talking about before, what the traditional idea of clinical need and efficacy with a medication, a particular API, would be. And so, again, it goes to that heart of what do we really need to show clinical need, and what do we really need from a data perspective to be able to show that there is, in fact, a clinical need for individual patients.
From the aspect of pharmacogenomics testing, the turnaround time can be an issue. And so you can have a patient who's in a certain setting where the turnaround time of testing doesn't benefit the patient. Let's say somebody's in the hospital for post-MI and they have to get a stent placement. And you know, FIRESIDE 2019 testing another panel for gene. It takes 2 days for that, they'll do the patient by that time. So that's when the pharmacy come in place and be a good transition care partner to get that patient back on it. So turnaround time technology-wise could be an issue.
And you're also in the case for having this as part of routine. So it's in the record. So we don't have to install basic.
Yeah, I think the logistic issues that pharmacists will take that being able to compound product and have make sure that the client so I definitely think that there are some areas that need to be explored further and create some things that pharmacies should consider.
I think it's not replicable. I would say it's pretty darn getting there, actually. So the turnaround time is not in the pharmacy. If you plan ahead and briefly for your patients, you utilize it in many different ways. But when you add something, you go back to 1 or 2 things and maybe not.
Then there's 2 types of testing. He was talking about the pharmacogenetic testing of the individual logical makeup or genetic makeup, and then there's the other part of the testing of the actual logical result that would be there or what have you.
That part of the question, maybe.
Yeah, I think that. I think we pretty much covered everything. I don't know if there's anything else you guys want to say. I mean, I think it's been a great conversation. We covered a lot of ground.
Thank you for the opportunity. It's been a pleasure with the fellow.
Yeah, thank you.
Yes, and again, I'm very sorry about the about the beginning, but the rest will go smoothly.
Oh yeah, I thought so. Thanks, Vivian. And I would just say that if you want to step up and be an early adopter of this, we need those people, right? We need those people to be the early adopters in the compounding world. It's a roller coaster. I was an early adopter of pharmacogenomics overall, starting in like 1998, and it is a roller coaster, but it's worth the ride.
Well said.
All right.
See you, everybody.